My name is Zuo. I'm a postdoc researcher at SGDP Center and a member of the crew. I'm very happy to host, to co-chair this session together with Professor Ian Campbell. And you will hear 3 talks with the theme of recognizing and reducing risk. First of all, let's welcome Professor Sylvane Desrivières. Her talk is multimodal risk factors for eating disorders, evidence from the imaging and astral cohorts.
Thank you, Zo. Please don't be too harsh on me in giving time. I can see the pointer to change. Oh, that's me. Okay. Thank you very much, Ulrike. I'm very, very excited to kick off this session. But on the other hand, I'm so humbled after this historical introduction that you made in eating disorders in the area and at the AOPPN, which makes me feel so proud now to be part of the team. Yeah. We have 10 minutes, and thankfully Matthew and Ulrike has already given an overview of eating disorders, so I'll keep the introduction short. But in trying to identify risk factors for eating disorders, there are key points that we keep in mind. The fact that eating disorders are really common, and prevalence of these disorders like anorexia nervosa, bulimia nervosa, and binge eating disorders increasing every time, even so after the pandemic. There is also this adolescence age of onset of most eating disorders, and there are really a complex presentation of those disorders with high degree of comorbidities with disorders like affective disorders, depression, anxiety, ADHD, and addiction. Already mentioned is also the high mortality associated with eating disorders. We all know that key to treating eating disorders is really starting early, with early prevention, diagnosis, better treatment, and understanding comorbidities and comorbid illnesses. But we just know too little about eating disorders, and we also lack reliable biomarkers in order to improve predictions. Yeah. So this is basically what we aim in our research, to identify early risk and resilience factors and biomarkers for eating disorders, and also understand better how those disorders develop, yeah, in order hopefully to identify new targets for interventions. And we do that in our group by using cohort studies that are really what we call multimodal, so that involves large type of data. And here are 3 of those cohorts. First one on the top is the Imagine cohort. That is an adolescent cohort, a longitudinal samples where adolescents were recruited initially when they were age 14, then followed up at age 16, 19, age 23. And you can see here that we collected a large amount of data from those participants, so including behavioral and neuropsychological data, clinical data, and a whole range of biological data, including genetic data, genotyping, epigenetic data, transcription, proteomics, so a whole battery of biological data, and also finally neuroimaging data. So this population longitudinal cohort is complemented by clinical cohorts, 2 clinical cohorts of patients with eating disorders, so anorexia nervosa, bulimia nervosa, binge eating disorders. So this is the ESTRA cohort. And the stratified cohort that includes participants with major depressive disorders, alcohol use disorders, a few cases of psychosis, and ADHD. The essential key here is that all those cohorts, Imagine, ESTRA, and Stratify, were assessed using exactly the same standardized assessment protocols. So we have used these cohorts, and here in particular, starting with the Imagine cohort, to study eating disorders and their development during adolescence. What you see here is the prevalence of eating disorder behaviors, binge eating, purging, and dieting across adolescence in Imagine. 2 very talented postdocs, Lauren and Zuo, have used this data set to try to understand the development of eating disorders. What they did actually is using the longitudinal data to identify a group of participants who would what they call developer groups. So they did not have eating disorder symptoms at age 14, but they reported eating disorder symptoms at later ages, age 16 or age 19. So they compared these developers groups to controls who had no eating disorder symptoms at any age. We have already published risk factors that predicted the development of those disordered eating behaviors, but today in this presentation, I will focus on brain and blood-based biomarkers. So first, brain-based biomarkers who are here, it really identified brain differences in brain structure that predated the development of future disordered eating behaviors. So what you see here in this slide, on one hand, differences, so higher volumes in the right putamen and globus pallidus, who occurred before the onset of binge eating disorders. Again, I can see the pointer in the top right panel. Yeah. So participants would develop future binge eating symptoms had already 5 years before onset of those symptoms, larger volumes in those subcortical areas highlighted in red. On the other hand, the development of purging behavior was characterized by lower volumes in the frontal cortex, highlighted, in the medial part of the frontal cortex, earlier before onset of those disorders. The interesting thing is that when you also compared the development of future depressive symptoms, the same brain area in the medial frontal cortex that was related to the development of purging was found to predate the development of depressive disorders. Another current PhD student in the group, talented, fabulous student, followed up those studies, now looking whether those differences in brain structure could be validated also in the group of clinical participants with anorexia nervosa or bulimia nervosa. So comparing, for example, participants with anorexia nervosa to healthy controls in the upper left panel of the slide, you can see highlighted in red a brain area that show lower volumes in anorexia nervosa compared to healthy controls. She did additional analysis. I will not go into detail, but one thing that we realized is that the brain area that she identified as being lower in anorexia nervosa partially overlap with the brain area that we found in Imagen to predate the development of eating disorder symptoms. Moving forward, we're also interested in comorbidities, the fact that eating disorders and other mental health symptoms are also comorbid. So now in this more recent study, combining the use of Imagen, Estra, and Stratify, the question here was whether participants with higher risk for mental health symptoms, including depression, ADHD, anorexia nervosa, bulimia nervosa, whether their brain structure or function would be different compared to healthy controls. And what you see highlighted in these are brain areas that really differ into those high-risk participants. And those brain areas are mainly localized in the prefrontal cortex, and they are also characterized by, in the panel C here, by areas that are interconnected and showing higher levels of interconnectivity in those higher risk participants. So this neural network, what they call it as a neuropsychological factor or NP factor, is a type of brain activity network that characterize participants with higher risk of mental health disorder. And this is related to executive control and executive functions. Importantly, this NP factor was validated in our clinical cohort of participants with depression, alcohol use disorders, and eating disorders, anorexia nervosa and bulimia nervosa, as being higher in those disorders. So is this NP factor an endophenotype for mental health comorbidities, and could that be a target for development of novel therapeutical interventions. 1 minute left. I don't want to end this without talking about blood-based biomarkers. We have already published, and this is in collaboration with our involvement in a large emerging genetic consortia, the ENIGMA consortia, that we can identify from blood epigenetic, a type of marker, epigenetic marker that really relate to brain and behaviors. So we decided to continue this work. And what Xinyang did here, she looked at Imagen data set age 14, whether we could identify DNA methylation patterns at age 14 that would relate to psychopathology in adolescents. So she identified different DNA methylation clusters. And here you can see 3 of those DNA methylation clusters and their associations with psychopathology during adolescence. All 3 clusters associate with pubertal development and associate with a range of mental health symptoms largely related to anxiety, depression, and importantly, eating disorders at age 14. Also relates two of those 3 clusters to personality traits, specifically neuroticism, which is known to be shared in eating disorders and other comorbidities. The interesting and important thing is not only she identified blood-based DNA methylation patterns, but also genes that are affected by those DNA methylation patterns. HOXC4 and OXA5, those are members of a gene family that really control development and systems development and organ development. In particular, OXA5 controls the development of the mammary gland. So these were interesting findings. She did also other studies to relate those DNA methylation patterns to differences in brain structure, and she found that differences in these DNA methylation patterns related to the OXA5 gene associate with reduced volume in subcortical areas, notably caudate and putamen. And here, the stronger the association with those subcortical brain areas are, the stronger their associations also with pubertal development, and all those differences related to the OXA5 gene are highlighted in red here. To finish, we are continuing our work in this worldwide cohort. We have continuing our epigenetic, cortical, and subcortical genome-wide association studies involving so far 17 cohorts worldwide with a sample size of over 8,000 participants. And we are currently just doing what we call meta-analysis to identify DNA methylation patterns that relate to subcortical and cortical brain areas. And we hope to have exciting findings very soon. I would like to finish this here by thanking really my team here. And I'm sorry, Diego is not on this part of this picture. Our collaborators and you for your attention. Thank you.
Take questions from the audience. I think we'll start with our audience from the front row. Please feel free to ask. Any questions? Please.
Where's the mic? Over there. Yes.
Thanks. Thank you for the talk. You said that there were changes in brain volume up to 5 years before the development of disordered eating behaviors. I think 5 years is like a very long time. Do you think there's a direct mechanistic link between these changes in disordered eating, or just sort of psychiatric conditions in general?
So we found that the brain differences predate, so come before even the onset of eating disorder symptoms. We don't know yet mechanistically, but there is the hypothesis that this is really a biomarker that can help to identify our future development of symptoms. What we're really trying to do also with Xinyang's work is try to understand the consequences of those differences in the brain in terms of behavior. You know, for example, Shinya has showed that those differences in volume relate, for example, to the cognitive restraint aspect of anorexia nervosa. And this is related to specific brain areas in the brain. So trying to identify brain structure that would mediate or explain disordered eating behaviors before even those these behaviors kick off.
Any more questions? Okay, we can discuss more at the break. Let's give another round of applause to the